an overview of how you might work in a lab situation with poliovirus, for example. We do have polioviruses in our freezer, but no opportunity to work with them. So anyway, the virus, as you see here, is a naked icosahedral virus, and it has a plus strand of RNA as its genome. And so one of the approaches to working with this is you could actually directly take polioviruses and apply them to a cell culture and then get these cells to propagate your polioviruses and collect them and do work with those. You could also take isolate the viral RNA from these viruses and transfect that into the cell. So essentially what that means is applying this material to the to the liquid here, and basically getting that liquid to be taken up, or getting that RNA to be taken up by the cultured cells in a process called transfection. This is safer, obviously, if you start by lysing your viruses and extracting their RNA. It's obviously safer because there's less of a chance for the workers to get infected. But obviously, on the back end, your goal is to propagate more viruses, and so you're still working with the viruses, but it minimizes your exposure on the front end. And so, again, in this particular case, you can convert this RNA to cDNA, to complementary DNA, and then create little virus plasmids. And then these plasmids can be genetically modified. You can do all kinds of genetic modifications to study different poliovirus genes, for example, and what their effect might be on replication. So this poliovirus DNA can be transfected into cultured cells as well, and then that can allow you to propagate. And you can also do in vitro RNA synthesis and convert this back into RNA, and then this RNA can be transfected. So after you've done your genetic modifications, you can do an in vitro RNA synthesis and then get your new plus-stranded genome with its modifications and do the transfection this way. So one of the big advantages of this, I found, is that working with DNA is much easier because DNA is more stable. RNA degrades pretty quickly in its free naked state. There's a lot more RNA digesting enzymes floating around in people's hands and on equipment than there are DNA nucleases. So this is just a little bit about constructing retroviral vectors, or working with, and how we might work with viruses like retroviruses such as HIV. And so in this particular case, you have here's a capsid plasmid, this plasmid basically, so imagine it like this, a capsid plasmid that has some of the main genes for constructing the plasmid, the group antigen and so forth, for making virus plasmids. And this other piece of genetic information here has different envelope genes. And so in these particular cases, the combination of two pieces of DNA, when they are transfected into these cells, will allow for the production of a virus without a viral genome. And the reason for that is the virus, these simulated virus pieces, these plasmids, lack the packaging signal. So the virus won't package this genetic information. It'll read it and ultimately translate it into proteins, but it won't package the information. And so that you can end up with a virus that doesn't have a genome. And so hopefully you can see why this would be advantageous. You can then use this as a vaccine. And so one of the cool things about that then is this vaccine would have no hope of making you sick. So it's a pretty safe way to make a vaccine. Another thing that you can do with this really cool approach is you can actually use this as a drug delivery system. If you know that this virus has very high specificity for binding to certain cell types, particularly if you can engineer these little spikes to bind to cancer cells. So imagine that you could have a way to package these viruses or build viruses without a genome, but then the viruses can contain certain chemicals, for example, anti-tumor drugs. So there's lots of different applications for this type of approach. And the main reason why it works, as I said, is it doesn't have this little packaging signal. The DNA doesn't get packaged for that reason. And so that's what happens down here. You've got capsid and envelope plasmids that will allow you to create full-on virions. And then you can insert any kind of genetic information you want on a third plasmid, as long as that third plasmid carries the upstream packaging signal. And so when we transfect these pieces of DNA into the cells, then a fully formed virus will be made and will be packaged with whatever is included here. And so again, the idea is that you can deliver even, for potential gene therapy. So, if you have whatever cells that have no longer able to produce insulin. Imagine if you can find a virus that specifically infected insulin-producing cells, and then you can insert the insulin gene using this type of a method. So some of the cytopathic effects. Cytopathic effects are evidence of viral growth in cultured cells, so we call them cytopathic effects. And these can be, you look at the cell culture under the microscope and see an altered shape, or sometimes the cells are just damaged so much they lift right off of the substrate. And so again, what I'm talking about is, in this particular case, imagine these are infected. If the cytopathic effect is severe damage, these cells can be lifted off and just kind of migrate away from the artificial matrix that they're attached to. Lysis is a big one, where the virus outright just destroys the cell and it pops open. Membrane fusion is another, where the cells, like I said, they form abnormal bonds to each other, and the membranes kind of just melt together into giant cells that are functionless. And then membrane permeability, like punching holes in the cells, and the cells start to leak. Inclusion bodies, as the name suggests, are these sort of really dense bodies that form inside the cytoplasm, and usually they can just be globs of virus pieces or things like that. But either case, it just makes a mess out of the cell interior and can actually kill the cell, because having all this garbage build up inside the cell is not good. Apoptosis is just a fancy word for programmed cell death. So the cell recognizes that it's infected by a virus and then will instigate a cascade of reactions to sort of kill itself. Not sort of kill itself, to actually kill itself. And so here's some of the cytopathic effects in action. And so in this particular case, you're looking at uninfected cells in one here, and you can see that they have sort of a classic teardrop appearance, and they used a dye that binds to and illuminates the cell membrane, so you can kind of see their outline. In step 2 and 3 and 4, as you can see, these are poliovirus infected cells, and some of these cells are starting to round up and lyse. And so you can see some of these little cells that have formed these little circles. They're starting to round up because the virus replication is damaging the actin cellular structure and so forth. And then eventually, even more rounding, and you can clearly see that there's a severe loss in density between this slide and this slide. And then ultimately, lysis, where there's just piles of cell debris and very clear areas where the cells have been cleared out. Another thing that can happen is the formation of syncytia. Syncytia is a fancy word for membrane fusion. So in this cartoon here, you can kind of see what happens. Here's 5 unique cells, that are very distinct from one another, but because the viruses have infected them, and then the virus has punched its little certain proteins through the cell membrane, these cells are now expressing virus proteins that link together. And then once they link together, they're pulled together closely, and then they form a big syncytium. And now you see all the 5 nuclei all floating around in the same cytoplasm. And that obviously kills the cells as well. And so these arrows point to syncytia in this particular murine leukemia virus infected cell culture. And obviously, if these are cells of your brain, for example, that would obviously cause brain damage. One of the things that we do is plaque assays where we use the cytopathic effect of lysis in order to determine the number of infectious viral particles in a sample. And so one of the ways you can do that is create dilutions of virus and then plate those dilutions onto lawns of susceptible host cells, and then you count the number of plaques that form. Plaques are areas of lysis, of viral lysis, and so hopefully if you took micro 224, you had the opportunity to do some plaque assays using T4 bacterial phage. And so we use the same kind of formula as we would for doing CFU counts, colony forming unit counts. So here we're using these plaques to do plaque forming unit counts per mil. So in this hypothetical example, then we've got 5 plaques on this particular plate. So we would put the 5 here as the numerator. The volume that we plated in this case is 0.2. So 5 divided by 0.2 times 1 over the dilution factor. So our dilution factor in this case is 10 to the minus 10. So that would be 1 over 10 to the minus 10. So now you can do the math and get a sense for what the virus titer would be in PFU, or plaque forming units per mil. So here's a lytic bacteriophage life cycle, and here's just some pictures of different kinds of bacterial viruses, just to give you the sense that there's a different range of morphology. So here you have a various icosahedral capsids, and then some with tails of different complexity, and then some non-tailed viruses as well, just to give you a sense for how diverse bacteriophage can be. And so here's a bacterium with a whole bunch of viruses stuck to it. And it's really neat to see this because it looks like the viruses are clearly honing in on a certain part of the cell, which suggesting that the cell only expresses receptors in certain locations. So they bind, and at some point they inject, relatively quickly after binding. It looks like a bunch of darts in a dartboard, but they inject their genetic information. And then the genetic information, one of the things that it does is it rapidly encodes enzymes, which would break down the host chromosome. The next thing that the virus does is then these viral DNAs in this particular case start to encode pieces of the virus. And so the viruses start to replicate and ultimately to assemble. What's interesting here is that you see some of the viruses have picked up, so imagine these blue pieces are the host chromosome that have been chopped into bits. And then these gray pieces are the actual viral genomes. You notice that some of these viruses have picked up host chromosome pieces. And then when this virus ultimately lyses the cell and the virus is released into the world, some of these guys, as you see here, have pieces of the host. And so this virus can go off into the world and infect something else. And that something else will not die and will not become a virus factor. It'll actually incorporate this novel piece of DNA that it acquired from the previous host. So here's a look at the different life cycles of viruses, lytic versus lysogenic together. And so we follow these things clockwise here. And so in this particular case, the phage has injected its DNA. The DNA integrates, enters the cytoplasm and circularizes in this case, and then serves as a template for making more virus. And then the viruses lyse out into the world. And so that's the typical lytic cycle. In many bacterial viruses, that lytic cycle would take 18 to 20 minutes, pretty quick. This is also the lytic cycle that can happen in certain human infections too, like herpes viruses can go lytic, and you see that as the cold sores, for example, or herpes lesions on genitals, for example. So another thing that can happen, though, is this lysogenic cycle. So imagine the viral DNA in this particular case circularizes, but instead of going into this phase, it actually integrates and becomes what's called a prophage. As a prophage, it can stably replicate along with the bacterium or host cell. And so, again, chickenpox viruses, which are herpes viruses, can do this really well. They get into an individual, and typically chickenpox will do this, and you'll get the chickenpox characteristic lesions. But some proportion of those viruses will do this and hide in your neurons. And so as a result, they stay quiet for long periods of time, and then maybe 30, 40 years later, they bust out and will go into the lytic cycle, and that's where you get shingles, for example. Or if it's a different kind of herpes virus that causes cold sores, the virus will do this and cause cold sores, but then eventually the cold sores go away because the virus has done this. And often the viruses that cause cold sores will just do this. Will just kind of quietly hang out. But then if something happens, you get super stressed out because you're studying for exams or whatever. You get too much sun one day at the beach and you burn yourself. The viruses are very sensitive to that, and they can actually bust out, sensing you're sensing that their host is stressed, and that will trigger a lytic response. And so the lytic response on plates, like I said, often looks like this, little plaques on a lawn of host cells. So all this dark stuff is a confluent lawn of host cells. And all these clear spots here are where the host cells have been destroyed. And they show up as physical spots on a plate.